Hello, everyone. Welcome back to the AAS YouTube channel. And this is part of the good stuff. This is the AAS Journal Author Series. And I'm super happy to have Morgan Cable with us today. Hey, Morgan.
Hello, how are you doing, Frank?
I am doing well on this November 1st, 2021. So one day after Thanksgiving, or Thanksgiving, Halloween in America. Thanksgiving's coming up. And where are you located, Morgan?
I am just outside of Pasadena, California.
Oh, very nice. Very nice. So usually that part of Southern California kind of gets their best weather, in my opinion, October, November. So hopefully things going well there.
Yeah, I hear some places this like frozen water falls from the sky, but we don't get that here. So it's quite nice.
I'm in Phoenix. Frozen what?
Exactly, yes.
You have some pretty awesome posters over your shoulders there. Those are very cool. Looks like one is Titan and the other is Enceladus.
Yes, very good eye. Yeah, those are part of the planetary exploration poster series that I believe Tyler Nordgren is the artist for these, but NASA's also put forth some other planetary exploration posters for exoplanets as well as places in our solar system. And they're really neat. You can download some of them for free. I think if you just go to nasa.gov and go to Travel Bureau or Travel Posters, you should be able to find them.
Yeah, I use them in my introductory planetary science course, and they're a big hit when people see them. So for the first time, it's pretty cool. Look like you got a nice old school NASA shirt there.
Yes, I do. I think it's really important to represent NASA. There's all sorts of cool swag that you can find online. I'm also sporting my Cassini jacket.
Oh, you are fully outfitted for this.
Yeah.
That is so awesome. Very cool. Very cool. So speaking of all that NASA stuff, and you're in Pasadena. So Morgan, what do you like to do for research?
For research, I do a couple of things, mostly focused around trying to understand organic molecules and search for biosignatures in our own cosmic backyard and beyond. Sometimes I'll be in the lab doing experiments to simulate, say, a Titan lake. And try to understand what kind of chemistry can happen there. And then sometimes I get really lucky and I get to go out and do field work at some places that we consider analog environments, to places like Europa or Mars or things like that. So it's a lot of fun, and I really enjoy helping to contribute to some of these really big questions about the distribution of organic molecules and the potential for life elsewhere.
Awesome. And that is going to bring us to this very lovely Planetary Science Journal article, The Science Case for a Return to Enceladus. And Morgan, take us away.
Oh, okay. Well, I was really excited to get the opportunity to work on this, and I want to give a massive thank you to all of my co-authors, in particular, Carolyn Porco. We worked together tirelessly for a while to get this paper into the form you see here. Now, this paper actually started off as a 7-page white paper for the Planetary Science and Astrobiology Decadal Survey. So this is one of, I believe about 500 science white papers that were submitted to sort of call attention to various things that we felt were important for the members of the panels of that decadal survey to be aware of. And so the whole point was to remind them and the science community that Enceladus, a small moon of Saturn, is one of the most fascinating places, in our opinion, and should be prioritized as a place to explore in the following decade. And so all of these co-authors contributed to this work where we build off of the discoveries of the Cassini mission, which was the most recent mission to the Saturn system, and establishing what we currently understand about Enceladus, its surface, its interior, its ocean, and the habitability of that liquid water ocean. And so if we step through this, we first talk about, yes, that ocean. Currently, the criterion for a habitable environment include liquid water, chemical building blocks, and energy. So first we wanted to address that liquid water point. Luckily, we have a whole host of evidence from the Cassini mission indicating not only that there's a global subsurface liquid water ocean, but that it seems to have been present for a while. We know a few things about that ocean. We know that it has salts like sodium chloride, sodium bicarbonate, and a little bit of potassium in there as well. And that's all from sampling of the plume. The Cassini spacecraft was able to fly through that plume multiple times,
Right.
had a couple of mass spectrometers that could look at the gas component, the vapor, as well as the grains, the ice grains that, they start off as liquid, but then when they hit the vacuum of space, they freeze. And so at the altitudes where Cassini was flying through, those were ice grains at that point.
Nice.
Yeah, and so if we keep going down, you can actually see these are a few of the first and the best images of that plume. So the one on the left is actually the very first ever sighting of the plume by Cassini. That was back in 2005. Then we've got another image that was taken with the narrow angle camera. So this is in the infrared, but it just gives you an idea of the structure of the plume. This is coming out of 4 big fissures at the South Pole that we call the tiger stripes. If you scroll a little bit to the right there, Frank, you can see this is an image taken, I think, at a phase angle where now you can see some of the reflection of those those ice grains off, thanks to sunlight coming in at a steep angle.
Nice, very nice.
And so we were able to characterize a lot of the properties of this plume with the imaging science subsystem, which you can see here, as well as those mass spectrometers and a few other instruments. So if we keep going, this figure 2 is our current best understanding of what Enceladus looks like on the inside. Thanks to gravity measurements, magnetometer measurements, and things like that, we have a pretty good idea of the thickness of the ice shell. We know that it's thicker at the North Pole, thinner at the South Pole, could be as thin as 5 kilometers or less. And that's important for us to know because it gives us some idea of how that material from the ocean is getting expressed into space, what it has to move through in terms of the ice shell to get there through fissures in cracks in that ice shell. We also have some idea of the thickness of the ocean. It's around 37 kilometers-ish thick, we believe. And thanks to the constituents in the plume, we have some constraints on the pH.
Right.
Now, this is something that's actually bounced around a bit in the literature as we have been able to better interpret and understand Cassini measurements. So Chris Klein, who's a colleague and a co-author, I think, on this paper. If not, he is. He's third author on this paper. Had published some work saying that, it may be more on the alkaline, more basic than this, around pH maybe 10-ish or so. And since that time has gone back and done a really in-depth look at how much hydrogen is in the plume, how much carbonation, so carbonates and CO2 are present, and how much methane is present. Now, it's kind of odd that we see both oxidized and reduced carbon in the plume, right? That's kind of weird. And that does tell us that there could be some heterogeneity in the interior, the rocky core of Enceladus, such that we can still have both of these species being generated. But based off of that, Chris Klein and others were able to better constrain that pH to fit the models that we have. And so 8.5 to 9 looks to be something that fits all of the data that we have so far. And this is important for habitability and for life, because we really want to understand what kind of chemical reactions would be favored under these conditions. And pH can drive some things to be either, delta G of positive or negative. And so that is important for us to understand if there are methanogens or other kind of organisms there, whether or not they could utilize those types of metabolisms to survive.
Yeah, nice.
A few other things here, hydrothermal vents, we have that constrained to interactions of 90 degrees C or above, so almost the boiling point of water. Sean Su and others have published some papers on that, and that's important because that tells us what kind of energy source this life could utilize. Remember, life needs liquid water, it needs organic chemical building blocks, and it needs energy. And so hydrothermal or geothermal energy could certainly drive some type of a primitive ecosystem here. we've got the tiger stripes and then the plume. And one thing that's important to note here is that these plume grains, you can sample them from flyby or from orbits from the plume directly, or you could sample them from some kind of a landed mission, right? Because about 93% of these plume grains fall back down onto the surface of Enceladus.
Okay.
And so that gives us a lot of different options to explore when it comes to mission architectures.
Indeed.
All right, let's keep going.
Very nice. Okay.
And thank you for driving, Frank.
Oh, my pleasure. And here we go.
All right. So let's skip down to this next figure. So this one also highlights some of those water rock interactions that could be generating hydrothermal vents. And it's important to note that this can lead to certain disequilibria in the ocean, whether those are redox gradients, pH, or other concentration or thermal gradients that life could potentially exploit. It's also important for us to think about how any molecules, any biosignatures, potentially if life is there, that are generated down at those hydrothermal vents, how those could be transported up the water column to the base of the ice shell, and then from there expressed through the plume.
Right.
That's something that's important for us to understand as well. And so keeping all of this in mind and trying to be conservative as we generate guesses for the types of life and the abundance of life that might be here are important things when we're designing future mission concepts to keep in mind.
Mm-hmm. Cool. Very nice.
And I should thank my colleague, Mark Naveau, who is another co-author on the paper who helped with this figure.
That is an awesome, awesome figure.
Yeah, they did a great job. So,
Right.
So we talked a bit about hydrothermal activity in the previous figures, but here's where it's summarized in the paper. So we've got a whole host of lines of evidence that support hydrothermal activity. The first is these silica nanograins, SiO2. You can see them on the top right there, right at the bottom of page 3. Sean Su was the lead author on a paper that utilized lab experiments to prove that, or to demonstrate that these kinds of particles of this size and this oxidation state only tend to form in very hot 90 degrees C interactions or above. Two other lines of evidence, if you continue going down, now we're on the left-hand side of page 4, are two gas measurements that were made by INMS, the ion and neutral mass spectrometer.
Yep.
One of those was for hydrogen, or excuse me, I should talk about methane first. One of them was for detecting methane. And the Bouquet et al. 2015 paper talks about this, that more methane was found than if it was had been trapped in, say, clathrates, those water cages, and released from some sort of mechanical action. So this suggests that it's being generated potentially by this hydrothermal activity at the seafloor. And then the third piece of evidence, so we've got the silica nanograins, we've got the methane, now we have hydrogen, molecular hydrogen, or H2. Now this one took a while. These papers are more recent. Hunter Waite and others in 2017 did an exhaustive study where they eliminated all other potential sources of hydrogen. Sometimes when water vapor gets inside of that instrument in particular, it can make some H2 just from interactions with the walls of the chamber. And so they were able to eliminate those as possible sources for the full amount of H2 that they observed. And so this is important because H2 is not only a product of hydrothermal activity, but it's also a great source of metabolism. It's essentially the easiest molecule to eat for microorganisms.
There you go.
And so that's important. Then we talk a little bit more about the pH, and I already covered that. So we included previous studies showing the wide pH ranges, and then report the more constrained pH range of 8.5 to 9 that is now consistent between two separate instruments, the INMS, the ion and neutral mass spectrometer, as well as CDA, the cosmic dust analyzer. Okay, so we've talked about 2 of the 3 ingredients for life as we know it, for habitable environment. We've talked about liquid water and we've talked about energy. But the last thing we need to talk about is these chemical building blocks, essentially the stuff that makes up the molecules that makes us be able to do what we do. And for biochemistry and biology, we tend to refer to that as SNOPs. Those are the main elements that you need: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur.
Yes.
And so we step through which of those have been detected with confidence in the plume and which detections are still a little bit tenuous. The ion and neutral mass spectrometer aboard Cassini did not have the mass resolution to distinguish between, say, CO and N2, which both have a mass of 28, for example. And so some of the detections are still a little bit tentative, but we have identified both carbon, oxygen, and nitrogen. So all three of those rich species.
Cool.
When it comes to sulfur and phosphorus, the story is a little bit more complicated. In this case, it's absence. What is absence of proof is not proof of absence.
Yes.
And a future mission could certainly be able to constrain those concentrations of those very important components to a much better degree. Again, Cassini was not meant to be a life detection mission. And so here we rely on models to constrain the concentrations based on assumptions of how Enceladus formed, how much leaching could be happening from its core into the ocean. And Tori Holer, who's another co-author on our paper, has done a lot of work on this. And so I encourage you to read a bit more of Tori's work when it comes to understanding the availability in particular of phosphorus.
Awesome.
So there's still some unanswered questions, but right now it looks like there are some equilibrium concentrations that right where you were circling suggest an average cell density of around 10 to the fourth cells per cubic centimeter or cells per milliliter. Could be supported. Now that's not bad. Earth's oceans are about 10 to 100 times more than that, around 10 to the 5, 10 to the 6, but there are some energy constrained environments, for example, some of these subglacial lakes like Lake Vostok in Antarctica that has around 10 to the 2 or 10 to the 3 cells per mil. So just to give you an idea, we're in a ballpark that could support detectable cell densities, but of course there are a lot of assumptions that have gone into that. And we talk about that in the paper. And I think if you go down, table 1 summarizes a lot of those. And we'll skip back up to figure 4. I do want to take a little bit of time to talk about that next. But if you would be so kind. Yes, perfect. All right, now this is a table that is reproduced in part from a book chapter that I was fortunate enough to write along with Tori Holer, Sean Su, and Mark Neville. And what it summarizes is based off of the energy flux that we assume from hydrogen in the plume, we can make some assumptions about what cell density that could support. In addition to that, there are some estimates of how many cells could make it into the plume grain. So going from the vent fluids down at the base to the bulk ocean to the plume grains. And so the main takeaway from this table is that there are a wide range of cell densities we might expect. They're based on certain assumptions that we include in these extensive footnotes that are larger than the table. But just to give you an idea, these would be the types of things that future missions might design to. And ideally, we would like these future missions to be able to handle even the most dilute cell densities and still be able to detect confidently whether or not life is there.
Cool.
So now let's go back up to figure 4. Now this one is something that I drew up based off of a lot of discussions with Frank Postberg, Nozer Khwaja, and a few other of our co-authors, talking about how different types of grains are generated in the plume. Now, not all grains are equal. We've detected a few different populations of grains. Some are very water rich. Those are type 1. They tend to be small and are almost pure water ice. Then we have type 2 grains, and these come in 2 flavors. One are silica rich, those nanograins that we talked about before that indicate hydrothermal activity. But some, about 4% of all of the grains that we see are organic rich, like 1% organic or more by mass. And that's really exciting because that tells us there could be some enrichment of organic material, kind of like we see here with sea spray aerosols on the Earth. You can get like this sea surface microlayer of organic stuff. And then when waves crash, it generates this aerosol that are similar in composition, organic rich and salt poor. So there have been some work trying to understand those grains. And then type 3 grains, these tend to be ones that are salt rich. So very rich in sodium chloride, potassium chloride, things like that, usually 0.5 to 2% by mass. And so this walks through a few different mechanisms for how these can form. Sometimes volatile organics can condense on the outside of those at the lowest temperature region, that shaded gray region at the top. But you can also have water vapor condensing on these as well in that purplish region, the narrowest part of the conduit. And so this just shows all the different ways that you can potentially make these grains that have different compositions that we see. And it's important for us to understand this because, for example, if I were to design a biosignature searching mission, I would want to hunt for those organic rich grains, right? Or maybe the salt rich grains, because it's possible that some species, like say free amino acids, might coordinate with some of the salts that are present. All right.
If you were, for example, to design one.
Yes, exactly.
So let me ask a question. This is just a really broad question. So what is it that is the main driver of Enceladus's energy budget? So it doesn't have, a molten core rolling around. So is it just latent heat of formation? Is it radioactivities? What is it that keeps Enceladus hot enough to do this?
That is a great question, Frank. We think the main reason here is tidal deformation. So Enceladus's orbit around Saturn is not perfectly circular. And that means that sometimes as it orbits, it'll experience slightly stronger tug or a weaker tug. And so the whole moon kind of flexes due to that gravitational asymmetry. And we believe that is generating enough heat. Now, there could be some radiogenic heating, that we can't rule that out completely. But as you step further and further out into the outer solar system, the concentration of those heavier elements tends to go down. And so I would say that radiogenic heating may be a greater or more likely contributor to something in orbit around Jupiter, like Europa, which also has a liquid water ocean. We can talk about that later if you'd like. But yes, the point is that our understanding of Enceladus's evolution over time indicates that that liquid water ocean should be long-lived. We also see this E-ring that is produced by the plume, and that's been around for a while.
Okay, cool. Thank you.
And actually, if you go down just a little bit, we are right into the part where we're talking about estimates of the age of Enceladus and its liquid water ocean that were previously debated, that have since been, I think, constrained pretty well. But there are still some lingering questions, and we talk about that here in section 5. One of the big questions is just, has Enceladus been around long enough, and has the ocean been around long enough for life to have emerged? And we only have a sample size of 1 for life, right? It's just Earth. That's all we know. And our best work still is confounded by the fact that Earth is very geologically active and has erased a lot of evidence from early in its formation. So the best we can do is say that life at least takes 1 billion years. It may be much, much shorter than that, but that's our upper limit on how long it takes life to form. And so it's possible that Enceladus's ocean has been around that long, but it could have been around for, say, 100 million years old. So there's still some further work that needs to be done, and more data of isotopic distributions at Enceladus's surface and in its plume and other things could help us address that to a higher degree.
Nice.
All right. Yeah. So let's keep going here.
Okay.
So we've already talked about this table. We're still outlining a few other ambiguities that can be nailed down. Certainly laboratory work is going to help with that. And now we get into strategies to search for life. So here we talk about different in situ measurements that you could perform, whether those are from fly-through or orbital science, or if you wanted to be able to land on the surface or even go down into the vents themselves and reach the ocean. And so here we do have one important table, which is a big one, but I think it's important to point out that a lot of science can be done from fly-through at hypervelocity. Now, this is what Cassini did, flying through Enceladus's plume. This is what the Europa Clipper mission will also be doing at Europa. And if indeed there's a plume there, as we have some tantalizing evidence that suggests that there might be, this could be a great way to look at organic molecules. Now, I and others have been actually working on this for a few years and wanted to summarize some of those recent developments here. So these are detections of different classes of organic molecules you might be interested in, for example, amino acids or fatty acids, and what velocities do these molecules ionize but not fragment, right? Because you want to be able to get them into the gas phase. This is the way CDA worked on Cassini, and the way SUDA, the surface dust analyzer on Clipper, is going to work. You have a metal plate. The ice grains come in, they whack the plate at 3 or more kilometers a second. And at those velocities, the ice grains go poof, they go into the gas phase, and they ionize as well. And this is perfect because now you can use a mass spectrometer to detect them. It's a really agnostic technique because it's not specific to any one kind of molecule. If it's got a charge, a mass spectrometer can see it. And so we and others have shown that 3 to 5 kilometers a second is really ideal for ionizing, but not blowing to smithereens these important biomolecules. And Fabian Klenner, who's also a co-author on this paper, has been doing a lot of work in this regard as well. And then Mark Burchell, who's not an author, but cited his work, has done some impacts with light gas guns. So a slightly larger than we would expect plume grains to be, but still really important work. And so I wanted to highlight those here. Yeah, the nice thing about these kinds of architectures for spacecraft is that they're a little bit less expensive because you don't have to land to do a biosignature assay, which is important.
Yep. Cost savings.
Yep.
Very cool.
It does matter. All right. And if we keep going, I think now we get to, yes, the point where we do talk about intact microorganisms, whether those would survive through the freezing process, being expelled by the plume for collection. Certainly at some of these velocities, if you're ionizing things, you're probably going to fragment or otherwise destroy the cells, but still detect the biosignatures within them.
Right, right.
Yeah. Now, if you collect slower, if you get into orbit and you're doing slower collections, then you could collect potentially enough material over multiple orbits to be able to sample cells. Or if you land, that's a great way to collect a lot of material. It's one of the benefits of landing. You can get orders of magnitude more stuff to collect and look for evidence of cells in particular, but other biosignatures as well. And so we felt it was important to essentially lay out the latest in our scientific understanding of Enceladus as a target to explore, both for habitability and life search missions, but also just as a really fascinating place to study, to see where different worlds lie on the habitability spectrum, how it got to be the way it is. And it's an important thing to bring out at this time because, of course, with the Decadal Survey coming out in March of 2022, this could be one of the locations that are prioritized with the future mission. It's definitely up to the Decadal Survey to evaluate that compared to all of the other attractive targets in the solar system.
Very cool. Very cool. All right. We good?
Yeah, I think that's about all I wanted to say. I encourage you all to go through and read this in more detail, but thanks for your interest. And yeah, if you want to talk about anything in particular, Frank, we can highlight a few other things, but that was the main point, is that when it comes to exploring a world like Enceladus, there are a lot of options, right? You can do a flyby, multiple flybys, you can do orbit, you can land. You can even work on some more exciting architectures to get down through those vents, right? You don't have to melt through hundreds or tens of kilometers of ice, right? You could even like climb down in there, and that could be an exciting thing for a future mission as well. So there are a whole menu of options when it comes to missions that could give us a lot more information about the habitability of this world.
Very nice, very nice. Morgan, thank you once again for walking us through your really lovely PSJ article. And you touched on it a little bit there at the end. So let me push on that a little bit. And that is sort of, like where does this idea go, let's say, over the next 5 years? So let's just, pretend and maybe say some decadal survey is super positive. Are there beginning motions to develop a mission? Would somebody be submitting a mission proposal? Is this sort of in the SMEX range, or is this in a larger range, or just, sort of where do we go over the next, let's say, 5 years with getting a mission to Enceladus?
That is a great question, Frank. So one thing I haven't mentioned yet is the New Frontiers mission call. So New Frontiers is in between Flagship and Discovery. The cost cap hasn't been announced yet for the New Frontiers 5 call, but we're expecting it to be somewhere between 900 million and maybe 1 point something billion dollars, which sounds like a lot, but really for a mission, that's a lot of bang for your buck. And I'm part of a team that is planning to put in a proposal to explore this amazing place. I think there may be another team as well. And so hopefully NASA will have a host of options to pick from. And then in addition to that, what you mentioned, if the decadal survey comes out and ranks Enceladus highly, potentially for, a flagship mission, that would be amazing. A colleague and a co-author on this paper, Shannon McKenzie, actually led a planetary mission concept study. This was something that went into the decadal survey evaluating a flagship concept for Enceladus. It was called the Orbilander, and there's actually another article in PSJ in this. It was either this special issue or one that came out just before or just after, which highlighted that mission concept. It would orbit, do a whole bunch of cool science, and then land afterwards and do even more cool science. And so I encourage you to check out Mackenzie et al. I think it's in the same issue, and check out that paper as well.
We'll put a link to that article in the description below this video. How about that?
That sounds great. Yeah, but the point is there are a lot of different things you can do depending on how you prioritize this amazing ocean world along with other amazing ocean worlds and other just cool places to explore in the solar system. So hopefully there will be something that goes to Enceladus in the next decade or so. That would be really fantastic.
That would indeed be fantastic, and we would need a new Travel Bureau poster to go along with that.
We absolutely would.
Very cool. Very cool. Morgan, thank you so much. And that'll do for this one. And I hope this made your astronomy day just a little bit better. And we will see you on the next one. Bye-bye.
Stay curious, everyone.